Prokaryotic versus Eukaryotic Cells, Part 2. When comparing prokaryotic versus eukaryotic cells, we find not only do we have no nucleus in a prokaryote and a nucleus in a eukaryote, but in the prokaryotic cells we would find no nucleolus, or plural would be nucleoli. There are no extrachromosomal sources of DNA in organelles, but instead extrachromosomal DNA in a prokaryote would be found in tiny little rings of DNA called plasmids. Cell division, or the reproduction of cells, occurs by binary fission in prokaryotic cells. In contrast, for these three characteristics I just described about prokaryotes, for eukaryotic cells, in a nucleus, we would expect to find, especially in plant and animal cells, a nucleolus, or more than one nucleoli, present in the nucleus of plant and animal cells. If we look at extrachromosomal sources of DNA in eukaryotic cells, we'd expect to find extrachromosomal DNA in mitochondria and chloroplasts. And for cell division, or the way that we reproduce more cells in a pro or eukaryotic prokaryote, would expect binary fission, though we have already seen some exceptions with that, with budding, in the examples in Unit 1, Chapter 1. But for eukaryotic cells, the type of cell division we'd expect to see in plant and animal cells particularly would be mitosis and meiosis. Now, there are a few protists that are eukaryotic in cell structure, but they do not use the very elegant, sort of sophisticated form of mitosis for cell division, but instead, many protists do use a form of binary fission. So cell reproduction for either pro or eukaryotic cells is not totally one or the other. It depends on the type of cell and how sophisticated that particular cell is. Now, in terms of extrachromosomal DNA, what does that mean? Well, as the name implies, extra means actually outside of. So, if we have DNA that's not included in the chromosome of a cell, then it's going to be called extrachromosomal. It's extra DNA, not involved in the structure of the chromosome, but found somewhere else inside of the cell. For a prokaryotic cell, it would be a plasmid. For a eukaryotic cell, it would be inside of an organelle like a mitochondria or a chloroplast. Prokaryotic versus eukaryotic cell membranes. The cytoplasmic or plasma or cell membrane that we find in a prokaryotic cell tends to have a fluid mosaic arrangement. Well, that's exactly the kind of arrangement that we see with eukaryotic cells. So it's not the sort of how it's constructed that's different between prokaryotic and eukaryotic cells. Instead, it's what it's made of that differentiates prokaryotic cell membranes versus eukaryotic cell membranes. Now, a phrase that I prefer to use when we're talking just simply about membrane structures is biological membranes. A biological membrane is typically formed from chemicals that we call phospholipids. We'll describe phospholipids in a lot more detail in Unit 2, but needless to say, that's a fundamental building block that's used in the construction of a biological membrane, including the cytoplasmic membranes or cell membranes. For prokaryotes, we find that oftentimes we don't find any sterols, like cholesterol, in the formation of a prokaryotic fluid mosaic cytoplasmic membrane. For eukaryotes, that fluid mosaic is typically made of phospholipids, similar kinds of phospholipids in prokaryotes, but the big difference in eukaryotic cell membranes, especially in animal cells, we do find cholesterol. The cholesterol is important to keep the cytoplasmic membrane of the animal cell fluid or very pliable. In prokaryotic cells, we find there are no structures that we would call endoplasmic reticula. If we're talking about singular, that would be reticulum. And the respiratory enzymes that are used in cellular respiration are found in the cytoplasmic membrane. In contrast, in a eukaryotic cell, especially plant and animal cells, we find rough endoplasmic reticulum and smooth endoplasmic reticulum. Both are used in the manufacture of the biochemicals the cells need to grow and repair themselves. The location of the respiratory enzymes that are used in cellular respiration in a eukaryotic cell are found on the inner membrane of the mitochondria in eukaryotic cells. Certain types of cells are capable of capturing sunlight energy and using a biosynthetic process called photosynthesis. If we have a photosynthetic prokaryotic cell, those pigments that are capable of capturing sunlight are chemicals like bacterial rhodopsin, or different kinds of pigments that are often located in structures that we call chromatophores. With a eukaryotic celled organism, the photosynthetic pigments tend to be chlorophylls, carotenoids, or xanthophylls, which are the greens, oranges, and yellow colors that we see in these types of eukaryotic celled organisms. We find the photosynthetic pigments are packaged in structures that we call thylakoids, and the thylakoids will be located in structures that we call chloroplasts, which are organelles found in plant cells. Additional specialized structures like lysosomes, peroxisomes, and Golgi apparatus are not present in prokaryotic cells. We can and do find them in certain kinds of eukaryotic cells. So lysosomes, peroxisomes, and Golgi can be present in a eukaryotic cell. Now, both pro and eukaryotic cells can have ribosomes. The difference between the two types of cells and their ribosomes is essentially the composition and the size. Now, ribosomes are sites or locations for protein synthesis. Protein synthesis is also called translation. So those are two different phrases or descriptions for the same process. The site of protein synthesis, also called translation, would be found on the ribosome. And for prokaryotic cells, ribosomes are 70S in size, versus ribosomes for eukaryotic cells in sort of the matrix outside of any organelle or nucleus in what we call the cytoplasm. The ribosomes are 80S, so they're larger than the ribosomes in the cytoplasm of prokaryotic cells. And for eukaryotic cells, there are other kinds of ribosomes that can be found. If we look inside of organelles, like mitochondria and chloroplasts, we do find 70S ribosomes. Now, the fact that the mitochondria and chloroplasts have ribosomes, and in fact, mitochondria and chloroplasts are also sites for extrachromosomal DNA. These mitochondria and chloroplasts can make copies of themselves, and in fact, mitochondria and chloroplasts appear to grow and divide inside of eukaryotic cells in this fashion that's very similar to the growth and reproduction or binary fission of prokaryotic cells that are independent or outside of, growing on their own. We're going to come back to this concept of the similarities between organelle growth inside of a eukaryotic cell and the independent growth of prokaryotes in the next section. Another distinction between pro and eukaryotic cells would be the cytoskeleton. There are no cytoskeletal structures in prokaryotes. You can find cytoskeleton structures in eukaryotic cells. If we take a look at the cell wall material, again, just as a reminder, the cell wall material of a prokaryote is either peptidoglycan or pseudopeptidoglycan. The cell wall material of a plant would probably be cellulose. A fungi, it would be chitin. And chitin, by the way, is simply, if you've ever stepped on a bug, that crunchy stuff that's used for insects to make their hard outer exoskeleton. So that same kind of sturdy material that insects use for their exoskeleton, fungi can use as part of their cell wall or outer covering. Additional differences between pro and eukaryotic cells. Prokaryotic cells might have a capsule or a slime layer on the outside of their peptidoglycan layer or pseudopeptidoglycan, whereas eukaryotic cells, if they have any kind of external layer outside of the cell wall, we would call it a pellicle, test, or a shell. Prokaryotic cells might have motility structures called flagella. Flagella are going to be made of a protein called flagellin, and in fact, eukaryotic flagella are going to be much more complex in their structure when we compare it to the very simple structure of prokaryotic flagella. The prokaryotic flagella is very similar to if you've ever had a mixing machine where you could mix dough, bread dough, and you have what I call a dough hook. So it's a very rigid helical structure. That's a very similar analogy to the rigid structure of a prokaryotic flagella. It's a very rigid, almost like a propeller or rotor on a boat. It's a very rigid structure composed almost entirely of flagellin protein, and in fact there are little sort of spinning discs embedded in the cytoplasmic membrane of prokaryotic cells that are responsible for turning this flagellum of prokaryotes either clockwise or counterclockwise. It's pretty much the only two directions prokaryotic flagella will go. They'll twirl or spin depending on which way the discs are turning. And think of the little discs in the cytoplasmic membrane of the prokaryote just like a record on a record player or a CD on a CD player. It has a little motor, a tiny little molecular motor that's going to spin the discs either clockwise or counterclockwise, and that in turn is attached to the flagellum and causes that flagellum to rotate clockwise or counterclockwise. If we look at the flagella of eukaryotic cells, far different. The flagella of eukaryotic cells are very flexible. In fact, they're composed of an assembly of what I call garden hoses, or microtubules. A microtubule, as the name kind of implies, is a tiny little tube. Again, very much like a long garden hose. We assemble those tubes together in pairs. We have, if we take a look at the image in the lower center of the slide, when we do a cross-section of a eukaryotic flagellum, we should see around just underneath the surface, on the periphery or outer edge of this flagellum, 9 pairs of microtubules. And then there's a central pair of microtubules right in the middle. Now what you can't see are the little tiny molecular motors that are hooking these pairs of microtubules together. And the movement of the microtubules is almost like a ratcheting movement, almost as if you're flicking your finger up and down, or lifting your knee up and down, lifting your leg up and down. It's kind of, again, that kind of flexing motion, and when you have hundreds and hundreds of these little molecular motors on the microtubules flexing up and down, that causes the eukaryotic flagellum to flex or twist just like a whip. In fact, the description of eukaryotic flagellum movement is, we say it's a whip-like motion. So, fundamental difference is, even though both pro and eukaryotic cells might have flagella, it's what they're made out of and how they move that's the difference. Prokaryotic cells and eukaryotic cells. Other kinds of different structures. Cilia, for one. You'll never find cilia on a prokaryotic cell. You may find cilia on certain types of eukaryotic celled organisms, especially the single-celled protists like Paramecium and Stentor. The cilia are used for motility. They're very flexible structures composed of clusters of long garden hose-like microtubules. The microtubules are very flexible and allow the cilia to easily bend from side to side. Notice that on the illustration of Paramecium, the cilia cover the entire surface of this little single-celled organism. Other kinds of structures that are unique to prokaryotes, but you will never find in eukaryotes, are the pili. Pili, also called fimbriae, are for attachment and conjugation. We'll talk about conjugation later in the semester when we talk about lateral gene transfer. But needless to say, the pili are short, tiny little projections that might be found on certain types of prokaryotic celled organisms, and again, they allow the organism to either stick to the surfaces or exchange genetic material. Some microbiologists reserve the word fimbriae for these same types of structures that are just used for attachment, and then they only use the word pili for those structures that are used for conjugation. Other microbiologists use the word pili for both kinds of functions, and they simply tell students which type we're talking about. Are they attachment pili or conjugation pili? And that's how we will use that word during the semester. We'll simply clearly describe what kinds of pili we're discussing when we go through the semester for the different topics. Another distinctive structure that we can find on some eukaryotic cells that we do not find on prokaryotes are pseudopodia. Pseudopodia are motility structures. They are not present ever on a prokaryotic cell, but for eukaryotic cells that do have them, pseudopodia means false foot. And of course, these are little extensions of the cytoplasmic membrane that allow certain types of cells to reach out, grab onto surfaces, and pull themselves along to move around in whatever environment that they're in. A typical kind of eukaryotic celled organism where we would find pseudopodia would be amoeba. Those are tiny little protists that use pseudopodia to move around in their environments.